the times the vaccines most of the time the vaccines are stable only at 2 to 8 degrees Celsius, and in most of the African countries and other places there may be unavailability of the refrigerations. So to avoid that, there are solutions like VVM labels available. These VVM labels are vaccine vial monitors which are being attached to the vials, and these have certain chemicals which will actually change their color if the vial is subjected to a temperature excursion. Because of this, the end person can actually identify that whether the vaccine has reached to them in like good condition or a bad condition, and then they can decide whether they can be used or they should not be used. Because if a subpotent vaccine is being used, it may or may not prevent the disease from occurring. So these are the different solutions that has come up, and these are available in terms of the preparedness for different pandemics. Now, let us understand what is the next generation vaccines versus traditional vaccines. Definitely, it is more safer because the risk of infection is less when we talk about any mRNA-based vaccine or a DNA vaccine or a VLP-based vaccines. They have a strong immune response because of the presence of the adjuvants. They can be customizable depending upon the scale and everything, and definitely they are more easier to scale up as compared to the traditional vaccines. So we have the risk of reversion in case of traditional vaccines, the limiting customization, and difficult to scale up. So most of the European and the American countries that do not allow us to sell the or basically supply the whole cell vaccines like attenuated vaccines and all that because of these sort of risk, although it is very, very less. And they are looking for mostly acellular sort of vaccines and all that. So, I mean, whenever we are talking about any preparedness, it has to be not only from the process point of view, from analytics, from all angles, it has to be there. So, we can focus more on questions. I think definitely you might be having questions related to this. So, we are open for questions.
Thank you, sir, for the valuable talk. And I request participant to ask the question in the chat box. And there is one question: how are researcher using CRISPR technology to develop new vaccine, and what are the potential implication of this work?
Okay. Uh, So CRISPR-Cas9 has been mostly used for the gene knockouts and all that. Currently, no commercial vaccine or any other vaccine which is in pipeline has been using CRISPR-Cas9 as such, but it is definitely in development with respect to getting the attenuation done for any specific whole cell viruses or bacterial vaccines. So that is only the application that I can envisage with respect to the CRISPR-Cas9.
So there is another question: how prime boost method is better than other classical methods of vaccination?
Right. So, means when we talk about the prime and boost, right? So the body also has like a capacity to develop the memory B cells, and what we have seen is currently, I will tell you a very interesting. I don't know most of the people know it about or not. Is the fractional dose immunization when we talk about the booster doses. So when we actually immunize, let's say first one is 10 microgram and then 10 microgram and then 2 microgram, we get a better immunogenicity as compared to when we give 10 10 10. So it means whenever there is a prime and boost, the definitely the memory cells actually are boosted up and you get a very high immunogenicity. However, people do not like to go for boost if there is like just one vaccine which can take care of that. Why? Because of the compliance part. because most of the time, if you have, let's say, 3 or 4 visits, then there are chances that they may or may not be able to comply, due to which you can actually lose out on the immunogenicity part. So there is again has to be a risk benefit ratio, whether we want to go for a good sort of antibody titers versus the compliance part.
So there is one more question. What is the current state of HIV vaccine research and what are the most promising strategies being explored?
Yeah, so for HIV is like, as you know, that even a separate organization, IAVI, was instituted for that, and many people were working on that platform. And most of the platform that is being used right now, including mRNA or DNA-based vaccines or VLP-based vaccines, everything has been tried and tested. But to say that have we got this solution yet, I don't think I can give any answer with that sort of a confidence. But yes, like most of the platforms are being utilized and they are being used, but nothing has been like you can say 100% efficacious as such.
Also, one question I have.
Sure.
So you said that preservatives are added in the vaccine.
Mm-hmm.
So can you just can you just throw light on how preservative is protecting the protein?
No,
Because
preservative do not protect the protein. Preservative is only for protection against contamination. Now, what happens is that whenever you have a multi-dose formulation or a multi-dose vial, the pharmacist may actually use 1 or 2 doses and keep the other 7 to 8 doses inside the vial. Now, because it has been compromised in terms of it has been used once or twice, the chances are that the microorganism or any other organism can actually enter into the vial and contaminate it. So we have to show that the preservatives are actually protecting any sort of a further contamination within a period of 28 days in which it has to be used. So that is how it is there. Otherwise, the preservative do not protect the protein.
so sir, you gave the example of some preservative. So
Hmm.
is that like broad antimicrobial
Yes,
against fungus
yes,
and bacteria,
yes,
or
yes. Most of the time, the anti also sorry, the preservative that are being used, like for example, thiamurcil, which is being used in most of the pentavalent and hexavalent vaccines, or the 2-phenoxyethanol, which is being used in most of other vaccines, is to get rid of both, because fungus also can grow and the other microorganisms can also grow, because that has to be immunized within a period of 28 days as per WHO. So it is a broad spectrum preservative, which actually, however, we have to actually look for the percentage that is effective in your excipient, because your excipients and the formulation buffer might change. So you have to actually optimize the percentage of the specific preservative that you are using.
all right, sir. So there's one more question in the chat box. How do nano vaccines work and what are their potential advantages over traditional vaccine approaches?
So when we talk about the nano vaccine as such, it is the what you can say, nanoparticle based formulations that are being used. Now, the only advantage with the nanoparticle based formulation is they actually are sometimes more stable, and they can have a more what you can say, shelf life as compared to the traditional vaccines. And most of the nanoparticles are again protein based. Now, once you have a protein and the other formulations going on, the immunogenicity can be compromised. So in terms of vaccines, we want to have a better immunogenicity. So in case of, let's say, once you actually immunize the vaccines, you want to have a sort of a decoy effect, so that the vaccines are not being flushed out from the body in a much faster way to get a better immune response or a humoral response. So these all sort of nanoparticles actually give a better decoy effect as compared to the actual live or the attenuated cells. And of course, the safety is better as compared to the attenuated or the live cells.
One more question is there, which shows higher side effects, mRNA or DNA vaccines?
So I think it will depend on what size of the mRNA or the formulations or the other things you are working with. Currently, what we have seen is every vaccine has its own sort of a side effects. But again, we need to work upon that. Okay, what are the those side effects, whether it is adverse events, serious adverse events, and to compare with respect to whether it is mRNA or DNA, I think it is a little absurd.
All right. So one question is, the vector used in the primary and booster dose in Covishield is same or different?
No, it has to be same only. It can never be different. So any vaccine that you work with, the vaccine platform and the deliverables, the drug product, everything is same. Only thing is that you are giving it in a different time period, so that I means it is primed and then boosted. But the all other formulations and everything is same.
Uh, so there is one more, two more questions. Is lyophilization preferred way to increase vaccine stability? And does vial material impact vaccine stability? If yes, then how we overcome it?
Right, that's a good question. Uh, the lyophilization is never preferred by the manufacturers. Why? Because the moment you have a lyophilized vaccine, again you have to give a diluent also. So it will actually increase the number of vials that you need to supply to actually immunize only one vaccine. So it will actually increase the load in terms of the number of vials to be shipped and the containerizations and everything, until and unless the protein is unstable enough that it will not sustain for minimum of 2 years or so also. So that is only the condition where some manufacturers might look for, no, I have to go for lyophilization because I cannot keep it in liquid form because of the stability issues. So first, any manufacturer will try to have all formulations tested to keep the protein in stable form. If they are still unable, then they will go for the lyophilization. But the moment they go for lyophilization, the cost and the number of vials that need to be shipped increases. So that is the situation. And second part was, if I recollect,
So
the
does vial material impact vaccine stability? Vial material.
right. So I mean, vial material. When I say the what you can say, the surface or the container closure, right? So the container closure definitely impacts the stability of the protein, because many times what we have seen is if the vials are siliconized, non-siliconized, the rubber stoppers are chlorobutyl or bromobutyl coated, or they are non-coated. So everything has an impact because of the leachability and the extractability. So all these things need to be checked before you actually end up with a final container closure.
So one more question. What about safety of mRNA vaccine?
So whatever the data that we have seen from the mRNA vaccines that has been supplied, we don't see any serious adverse events in terms of the mRNA vaccine. That is what the best I can say. Otherwise, I mean, the DNA vaccines are also available. mRNA vaccine is still going on. So I don't see any alarming situation where we should not be in future using it for any other pandemic.
So can I have a question regarding this mRNA safety, mRNA
Mm-hmm.
vaccine safety?
Yeah,
How
please.
how the vaccine is processed in the body?
So exactly, I am not looking after the mRNA vaccine as such. I mean, mostly I'm looking for the VLP-based vaccines. If you want to exactly understand how mRNA vaccine is being introduced inside the body or the safety profile, then I have to get back to you on that?
Yeah, that's I'm very keen about understanding
Okay.
because RNA is a live molecule and it
Mm-hmm.
might induce immunogenicity and give a protection for some time.
Mm-hmm.
But if the molecule remains in the body for a long period of time, then what is its fate and how it is being looked at? What are the in terms of the duration for which it has been examined and things like
So,
that. More
yeah, so
specific,
definitely I can get back to you if you can, like, I mean, you can write to me or so that I can give you a specific answer if you are looking for this.
sure. I'll get in touch with you.
Sure.
So there's one
Thank you.
question in continuation to the previous one where they
Mm-hmm.
have asked you about whether the vector will be same for Covishield or primary boost. So he's
Mm-hmm.
saying if vector remains the same, then immune system may clear it during secondary response.
No, it's not like that. Because when we talk about the vector or anything, even let's say if the proteins, right, or the VLPs, anything which is there, it, I mean, as you are saying that ki it will be cleared from the body, but the moment it is being cleared, it will also increase the like the antibody titers. So it is not like that ki we just change it. We keep the things constant, and because the safety profile has to be studied accordingly. And if there is the vector or any other thing is changed, again you have to study the safety profile of those vectors as well. So the vector that has been used in the primary that only can be used in the booster doses, and it definitely as you said ki it can be cleared off, but it will already enhance the immunogenicity to a certain extent before getting cleared. So that is what the best I can say right now.
Sir, we don't have any other questions in chat box. If any
Okay.
participant has, please write it. So just, I was interested, like Covishield was developed by Serum Institute.
Mm-hmm.
So can you
Mm-hmm.
throw some light on how did it happen and if you are part of that team or?
Right. So when actually COVID started, we prior to that we were working with these two organizations. One was Novavax, where we were working with the Matrix M adjuvant, and then we were also working with Oxford for the malaria product. Now, once and malaria, they have worked on various ChEDOX platforms and other platforms. Now, once COVID came, so we were trying to figure out which vaccine we want to take forward. And so we were working on multiple platforms, including mRNA, including DNA, including receptor binding domains. So out of all those different strategies, the Covishield worked better with better immunogenicity and better safety profile. And the chimpanzee adenovirus platform was working fine at that time, although we were not having that sort of a stability data. So therefore, we could only propose for maximum of 9 months of stability. We could not get time for increasing the stability profile also, because everybody was against okay, no, we want the vaccine at the earliest, and to get to a stability or the shelf life, you need to optimize certain conditions and excipients. For that also, we were not getting that sort of a time. Whatever the minimum stability we could offer, and because it was actually being supplied right away, so therefore also that didn't harm. Otherwise, we wanted to actually increase the stability from 9 months to maybe around 15 to 18 months. But that is how it worked out at that time.
So, sir, did you just replicate the AstraZeneca's model, or you like the Serum Institute
So
people refined some processes or steps?
yeah, so means the model of serum is like we actually get hold of the clone and we develop the process in-house. So when you talk about the AstraZeneca, the process, the system or the clone and the things were developed at Jenner Institute, Oxford, and it was actually taken by both AstraZeneca as well as Serum parallelly. However, AstraZeneca could not scale it up, and we could actually scale it up to a much larger scale. Actually, we stopped all other formulations and filling lines to actually supply only the COVID. So at a time, we were having almost 10 filling lines only manufacturing COVID. So we were actually working on the process development part, where we have increased the scale to around 2.5 kiloliters for COVID. Otherwise, AstraZeneca was not able to scale that up to that levels.
So in that case, we were more advanced than
I
them,
mean,
or
actually, Serum is the number one company in terms of the number of doses supplied, and that is because we have the scales and we have the expertise to increase the scales. So when you talk about even the malaria, now GSK is the competitor of for malaria, but when the UNICEF asked for around 250 million doses, they could supply only 10 million doses. We are supplying 200 million doses per year, so we can reach to that level of scales. That is what our expertise is.
all right, sir. That's very commendable and good to hear this. Uh, your talk was very informative, and certainly you answered all the questions. And I hope there is no other question. So,
Yeah,
yeah.
sure. Thank you all for patient hearing. And if you have any questions, you can definitely reach Dr. Shilpa, and she can like forward any specific questions if you people have. I would be happy to answer.
Thank you so much, sir, for taking out your precious time, and you accepted our invite. And thank you so much, sir. Thank
Sure.
you. Thank
Thank
you.
you. Yeah.
Thank you. So our next session will be at 7:30. I'll just post the attendance link here in the chat box. So 7:30, we will be joined by Dr. Anil Kaul. He will be giving talk on behalf of Sanjeev Navangal from BSV Group. And the topic is latest trends in biopharmaceuticals. So the meeting is on. You can fill the attendance form, and we'll start at 7:30. Thank you. So now
Are
your
you able
screen
to?
is there. Yeah, yeah, it is there, but your PPT is not there. We can see the
Yeah,
Zoom page.
yeah, that should be okay. Can you see it now?
Yes, sir, we can see now.
Okay.
So, but full screen you need to do.
Yeah, I will do the full screen. Yeah. So you want to have any introduction or anything?
Yeah. So I want to welcome you first, sir. I was just working on the logistics right now, but I'm glad that you have joined, and I know that you are uh, abroad. And thank you for consideration and accepting our invite to be resource person for this FTP. And so now I request my PhD student Twinkle to introduce you, our speaker. Twinkle, are you there?
Yes, ma'am, I'm here.
Yeah, please go ahead.
Thank you, ma'am. I feel honored to introduce our respected speaker, Professor Anil Kaul. He is a scientist and innovation leader with over 25 years of global experience in drug discovery, clinical development, and public health research. He previously served as vice president and of R&D at Johnson& and Johnson and was a member of the board of Janssen Pharmaceutical in Belgium. He was part of the team that discovered and developed bedaquiline, the first new tuberculosis drug in 45 years, now approved in more than 170 countries and listed by WHO as an essential medicine. His work has advanced multiple drug candidates into human clinical trial, and he has received several honors, including the American Chemical Society Prize 2020 for the discovery of bedaquiline. Professor Kaul has served as director of CSI IMTech in India and as a member of CSI Scientific Advisory Board. He is an inventor on over 35 patents, has published in leading journals including Nature and Science, and was recognized among India's top 20 bioscience innovator in 2023. He is currently an adjunct professor at the London School of Hygiene and Tropical Medicine and Group Chief Scientific Officer at Mankind Pharma, Bharat Serum and Vaccine, India, and served on board of several institutes and startups. With that, I now request Professor Anil to proceed with the session. Thank you, sir.
Yeah. So, Shilpa, should we go ahead?
Yes, sir. Stage is all yours.
Okay. Before we go ahead, I'm not sure of the context of this meeting and the audience. So if you can give me a background so that I can modulate the talk accordingly.
So, sir, the participants, we have the faculty members, assistant professors, associate professors from varied backgrounds. And the topic of this FDP, the Faculty Development Program, is Emerging Technologies for Healthcare Applications. So,
yeah, that's fine then.
Yeah.
So what we will do today, I know it was biopharmaceutical, so I won't be talking about biopharmaceutical today. We can do it at a later stage. So today,
Okay.
because of the time, we will speak on TB, which is, I think, it's very relevant to India. And we will try to give some not full context of role of AI and how we discover new medicines. It's mostly a story of drug discovery and how we discovered bedaquiline. But now with AI and machine learning coming into drug discovery, how I think it will change the future of drug discovery in India as well as across the world. Is that okay?
Yes, sir. Sure, sir.
So thanks again, Shilpa, for inviting me. My name is Anil Kaul. As it was said, I run a lab at London School of Hygiene and Tropical Medicine in London in UK. And I'm also the group CSO of India's one of the fourth largest pharmaceutical companies called Mankind Pharma. And I also head the BSV, which is a fully owned subsidiary of Mankind for all in terms of the innovative R&D and innovation. So I have been working with mostly all my life in pharmaceutical industry. Like I was the VP for R&D for Johnson& and Johnson, and I have been also working with a lot of public sector institutions like CSIR. I don't know how many of you know CSIR IMTech. So it's one of the India's very finest institution where I was heading that institute between 2016 and 2019. So today I won't talk about anything, as I said, that we will talk about bedaquiline. I won't talk to you about why it is important, but I will tell you when you do a discovery, what is the impact of that discovery on patients across the world, or when you develop a new medicine. Then I will talk to you about some of the latest research we have done in discovering this molecule 6640 and how does future of TB look like. And then finally, I think the last one is basically I keep on telling my aspiration of world without TB. So drug discovery is very serendipitous. It's very challenging, it's very difficult, and it's like finding what they call needle in the haystack. It used to be like that. But now with the advent of new technologies, things are changing a little bit. But at the same time, it's difficult because it takes almost 10 to 12 years from the discovery to approval of a drug, if you are extremely lucky. The cost is more than $1.5 billion to develop a new drug, and only one molecule out of 10,000 will come to the launch. I will skip this slide, but it tells you that there is the more the money comes into the science, the less we are getting in terms of the translational output of that particular science. So it's called Erome's law, which shows that exponential drop in the R&D productivity. That's why, as I said, that the cost of developing new drugs is really increasing exponentially, and it's not really profitable anymore to discover and develop new drugs unless until you increase the cost. TB is a big challenge. We all know that. We have 10.6 million infections every year and almost 1 million deaths because of the TB across the world. And out of that 1.5 million, almost 500,000 people die because of MDR tuberculosis, which is really the drug resistance form of TB. What does MDR mean? What does drug resistance mean? Drug resistance means you have drug susceptible, which is a TB, normal TB, which can be treated by rifampicin and isoniazid. Then you have MDR TB, which is resistant to rifampicin and isoniazid. And then you get into pre-XDR and XDR category, where on top of the rifampicin and isoniazid resistance, you have resistance to the injectables also, like amikacin, kanamycin, and capreomycin. These are the injections you normally give to the TB patients, you used to give to the TB patients. So then finally, XDR TB is basically where you have resistance to the entire gamut of drugs available, and it's really difficult to treat these XDR TB patients. So I can tell you this slide is very interesting for some of you. So this was a hospital, which is the MDR TB hospital in Banaras. And I visited that hospital and we met this guy, Professor Srivastava. He was the head of the TB division. And there was a case of 24-year-old male on MDR TB who had MDR TB for almost 1.5 years. These were his symptoms: cough, body weight loss, he had left the school, non-alcoholic and non-smoker. And he was put on the WHO convention regimen at that time for MDR TB. The family history was very tough. This tells you because the father and brother died while on treatment at XDR TB. The mother was recently diagnosed on the XDR TB. He himself had XDR TB, and in the family, only one person was negative for XDR TB. So finally, at that time, doctors put him on bedaquiline, and the hope was that he will be able to survive. So this is the bleak scenario of drug-resistant TB in India or across the world. It's not only India. So the lesson is that when there is something like this, where the chances of survival for these patients were less than 30%. So what do? The lesson is that when something is very important enough, you do even if the odds are not in the favor. The odds are basically the high cost of discovering and developing new drugs. TB is not a prioritized disease for most of the companies or most of the pharmaceutical industry, and there is very little money which goes into the drug development for TB. So this was a drug which we had discovered called bedaquiline. It was called TMC-207 or 207-910, and we discovered it through through inhibition of what we call ATP synthase, which is basically the target of this particular drug. So ATP synthase is an enzyme which basically synthesizes the ATP, and bedaquiline shuts down basically blocks this particular enzyme, and at a very nice IC50 of 2.5 nanomolar, and basically blocks the energy uh, supply to the bacterium. So this was the first experiment I did myself at that time, which was long time ago now. So we were still wondering how does this compound or drug kill the bacteria? So we did this experiment where we used this compound, which is basically the bedaquiline, and I linked it to the Cephalus column with a linker, and we created this compound resin. At that time, everybody said that this is not going to work. We are not finding any proteins which will bind to this affinity resin. And but to my surprise, we found only two proteins which were binding to this resin. Why this was very difficult? Because this molecule is a very lipophilic molecule, and as such, it doesn't bind many drug. It binds to a lot of non-specific proteins in the cell lysate. But upon analysis, we found that indeed it was binding to two main proteins, which is alpha and beta subunit. So the lesson is that don't listen to external noise, follow your instinct and the gut. If you believe that this thing is going to work, try to give it your best shot. So this is the ATP synthase where bedaquiline is bound. You can see that this is the enzyme. This was published recently in 2021 in Nature by a group in Canada, where they showed that this is the drug bedaquiline, and they showed how exactly this drug binds to this ATP synthase. So this is the C subunit of the ATP synthase. As you can see that it's a very, very complex machine involved in the synthesis of ATP. And this is the drug which is coming here and binding to this particular thing. And as it binds to this particular C subunit, the gray one in the bottom, let me put the arrow pointer. Yes. Can you see the pointer? Yes, I hope so. So this binds to it, and as such, you have the block of the ATP synthesis. So why is TB difficult to kill? So because TB is difficult to kill, because it is very heterogeneous population. Why is other bacterial infection easy to kill? Of course, there are several reasons for that, but one of the reasons is that because there is what we call heterogeneity. Heterogeneity means that in that TB population in a patient, there will be some bacteria which will be dividing fast, which is called alpha, and there will be some which are dividing slowly, which is called beta here, and then there are population which is the gamma population, which is basically the dormant. So what we found that bedaquiline was able to kill both rapidly replicating as well as dormant TB populations very, very effectively. So this was very interesting because no other drug had a mechanism of action like this, where they could kill both the fast as well as dormant TB very good effectively. Because why they are killing dormant TB? Apparently ATP synthesis is very essential in dormant bacteria also. So then we went to the clinical study, but the first clinical study was really a shock for us because it was not really effective. So this was what we call monotherapy. On x-axis, you have number of days for which we treated these normal TB patients, and on y-axis is the decrease in the sputum concentration of the bacteria. So you could see that the two control drugs which we used, isoniazid and rifampicin, were very effective, but bedaquiline in this particular study was weakly active, or active only during the last 6 and 7 days. So it was only 0.8 logs. So this was a surprise. We said that what the hell is happening here? Why we are not able to see is it because of the ATP pools and drug not going to the sputum proteins, time-dependent killing mechanisms, or metabolic adaptations? So lesson is that every project or every drug fails at least once during its life cycle. So you need to have faith and you need to have good science to pull it through, and it's what we call the development cycle. And persistence is the key. So this is an experiment which was done by one of my friends, and what he did is that he picked up these are the green is the GFP labeled Mycobacterium tuberculosis, and he put a single cell, what we call single cell time lapse video microscopy, where he, from a single cell, he could see the impact of Betaclin at a single cell level. So what you see is that during the first 4 days, this is an experiment of 24 days. You can see 400 hours. And during the first few days or 7 days, nothing really happens. But the kill starts from the day 7 onwards, and it's very nice kill after day. So this is exactly what we had seen in the clinical study was shown here. I will show you this video. This is the bacteria, TB bacteria, very nicely dividing. This is the normal media. Now the drug comes. Nothing really happens during the first 7 days. But after day 7, you see that the bacteria simply blast away. And that's where the impact of the drug was shown, that there is a delayed mechanism of action which will lead to the efficacy of the drug. So of course, then we did another clinical study of 2 months where we used this drug by this guy, Andreas Diakon, where he showed that the Betaclin, instead of 1 week, which we had previously done, we now did a trial for 2 months on MDR TB patients, which is all published here. And we showed that the drug was very effective in blocking the and this the in curing the patients of TB. Basically, the red line is basically the decrease in the bacterial burden in these patients, or the number of patients who are cured, kind of. And this is the Betaclin, and the blue line is basically the placebo. The drug, the placebo here. This is the placebo, and this is the drug. And you can see by 2 months, you had almost 50% uh, uh, of culture conversion, which was positive, and here it was only 10%. So, of course, a lot of data has been produced now, a lot of studies. I won't go in all these details to show that the drug is almost 90% effective in most of the studies across the world, in across 12,000 patients. So now, where are we right now? I'm just cutting the whole story short. We had the first TB drug with a novel mechanism of action, which was discovered. Estimated 5 million infections have been prevented till now. More than 1 million courses of Betaclin have been delivered. So 1 million people have been impacted by lives might have been saved because of the Betaclin. The drug is available in 170 countries. In 73 countries, it's formally approved as part of the regulatory guidelines. And now it's recommended by WHO as part of the all oral. I told you before that the person with TB or drug resistant TB had to go every day for 6 months to get an injection for treatment. But now with Betaclin, they don't need to go to the doctors or to the hospital. They can have all oral drug resistant TB regimen. And the medicine is now part of WHO's list of essential medicine for people with pulmonary TB as well as with children. So the challenge with the children is that we still have 1 million cases of TB in children every year. Very neglected disease in children. And what was done in May 2020, Bedeclin was developed as a pediatric formulation, was approved by FDA. So one of the last year there was a report in Times of India in Mumbai. There was a couple of kids, I think around 20, 30 kids, and who were given Bedeclin. They had XDR TB, and one of the kid was as old as 4 months. A 4-month-old baby having an XDR TB. So you can see that XDR is no longer because of the resistance, drug resistance, but because it is being spread in the communities, and kids as old as 4 months can have this disease. And then it shows that Bedeclin cures 4 kids of the drug-resistant TB. So really effective in adults as well as children. So now future is, as I said, that apart from Bedeclin, it's still used in combination with 3 or 4 other drugs. So what we are trying to do is develop new drugs which can reduce the treatment duration. And how do we develop new drugs is using the AI. So one of the key things in the AI is what we call generative chemistry. So if you have a target, without going through the screening platforms, you can generate new molecules, and these will be novel, these will be new chemistries, and these will be new. And that's where the whole field is moving towards, that without having those compound libraries, without going through the screening technologies, you can develop new drugs. Of course, you can also design new drugs if you have a chemical scaffold or a chemotype, what we call, through what we call AI ML model, using the augmented molecular design. You can also know the features of the drugs in terms of the off-target effects using the biosignature platforms, where you can basically visualize what is the impact on a normal cell of this particular drug, which pathways, and this is all done via just by image analysis, whether it is doing anything to the eukaryotic cells in terms of the uh, nuclear transport or any other cytoplasmic impact or things like that, so as to de-risk the cytotox very early on before you go into the animals. So now how the future of TB looks like, as I said, that it's all now all oral, 6 to 9 months. Now what we want to do is to reduce the treatment from 6 to 9 months to 3 to 4 months. And for that, you need new drugs in combination with Bedeclin, which are more potent, which are more bactericidal, which can kill very fast, and which have less side effects. I can skip this. As I said that, and how do we do it? Once again, I'm very focusing right now. I don't know how many of you are chemists in the background, or doing AI and the generative chemistry platforms using the deep reinforcement learning engine is very effective, not only defining the drugs, but also designing the drugs which are drug-like, like what we call, they will have features of admit, which are built into those molecules. And if it took us 5 years to bring a drug to clinic to phase 1, now it can go much faster. I will skip this. So using these platforms, we discovered several new molecules in the last couple of years beyond Bedeclin. And one of the molecules was, as I said, that is the ATP synthase. And we discovered this ATP synthase, and then we also discovered what we call new drugs, which are targeting the same pathway. So we are developing drugs which can work in synergism with Bedeclin. So, in that sense, we have been working on new drugs targeting the cytochrome BC and menaquinone. So what we call the carbon metabolism of the cells. So bedaquiline is in the end an energy inhibitor or metabolic inhibitor because it blocks the ATP, ATP being the central molecule in the metabolism. We were looking at what else can we block so that we can shut down the entire power supply of the bacteria. And and a lot of people said that, yeah, this is not the right strategy. This is not good strategy to discover new drugs. There's no synergism, but the lesson is that you have to think a little bit different than how the crowd, everybody else is thinking. That's where the new innovation will come through. So we discovered, using this strategy, we discovered several molecules, but one of the latest data I will show is discovery of what we call pure F. uh, a molecule which targets purine biosynthesis. I don't know how many of you know the basic biochemistry of purine biosynthesis is basically its purines are very essential for synthesis of nucleic acids and like DNA, RNA, they form the base of that nucleus. So if you block that purine biosynthesis, in bacteria or in eukaryotic cells. People have tried it in cancer cells also to target purine biosynthesis. They become very effective. Of course, the key is that if you develop any molecule, it has to be very selective to the bacterial biosynthesis rather than the mammalian. So we discovered this molecule called 6640, a very nice molecule with an MIC or killing bacteria, you can say bacteria of 8.6 nanomolar. So this all data which I'm showing here in the next few slides has been yeah, let me show how can I hide it. Okay, I cannot hide it. Has been published in Nature last year and this year only. I don't know why I can't move this. Yeah. So what we did is that we identified this. We showed that through various resistance selections. I won't go into detail that this molecule JNJ6640 is indeed targeting purine biosynthesis. We did what we call uh, the crystal structure analysis using the AlphaFold and showed that it binds to this particular pocket and yeah. Get stuck. Yeah, we did the CRISPR knockdowns to show that indeed this drug is basically blocking the pure F. And if we have the CRISPR knockout of the pure F, the bacteria are very, very susceptible. What was very interesting, I will not go in the metabolomics data of this. I will just simply show it's all published in, you can see we did a kind of what we call targeting metabolome to show that if you block the puref, there's a change in both adenine, AMP, and glutamate levels of the bacteria. So what is interesting is that, as I said, that purines are in humans also, and purines are in bacteria. So the enzyme in human is called HSPPAT, and we showed that across different cell lines, we showed that JNJ6640 was very specific to the bacterial and had no impact on purine biosynthesis across different cell lines. and the selectivity of the enzyme in the enzymatic assay for M. tuberculosis, pure F, versus the human PPAT proteins was greater than 10,000 fold. So very safe, very selective, very nicely blocking the purine biosynthesis. Uh, yeah, so I see that the eventual goal is what? The eventual goal, as I said, is basically blocking the bacteria in vitro in the combination with bedaquiline. As I said, that our aim was to develop a drug which can replace some of the old drugs which are currently in the regimen, like linezolid, with this particular compound. And this is the data here where we show that we can replace this. So, and we can replace linezolid very nicely. Linezolid is very toxic. It has an impact on your bone marrow cells. So we could easily replace it both in vitro as well as in the mouse model. So I'm just quickly going through it because I know there's a lot of data. The expression is basically that, can we have a TB as a single pill? Right now, it's a still combination of 5 to 7 pills, and the aim is, what the world is looking for is reduce the treatment duration to less than 3 months, and whether we can combine 2 or 3 different pills together into a single pill. And for that, what we need to do is that we need to identifying uh, missing millions of people who are still underdiagnosed with TB, expand the access, what we call the access framework, and advance the R&D. So beyond all the science, I think it's very important that the scientists talk to physicians, to the clinicians, to the patients, to really see where the unmet medical need is. So I was in the recently, I just showing you the one of my interaction with KGMC Medical College in Lucknow, and one of the patients who was receiving bedaquiline was was a girl of 20 years old who had spinal TB. So it's not only the TB is pulmonary, the TB can be extrapulmonary. It can go in your stomach, it can go you in your brain, it can go you in your spine. And that is also a big challenge, where we have almost 20 to 30% of cases of TB which are undiagnosed, is because it goes into the other organs. So a lot of partners, and I can skip this. This is the last slide to tell you that this was a story where I went to the South Africa, where I went to a hospital which were treating MDR TB patients. We were 4 or 5 people, and as we were walking in the hospital, we saw the pharmacy of the hospital, and there were all these boxes in that pharmacy, and we looked at it, was written BL across all these boxes. So out of curiosity, we asked them, what are these boxes? They said, this is bedaquiline. So this tells you the impact of a research where you do somewhere in the world, it has on the patients somewhere in the world where you may go or may not go ever in your life. So that's the power of the science. That's the power of what we do every day in terms of trying to use science to build innovation, medicines, treatments for very difficult diseases. So a lot of people in London School which have done the Pure work. So I'm not showing you the people who have done better work, because that's very large group. That's so many people. I'm just telling you the story in a very short, precise manner, but I can tell you there have been hundreds of people involved in the Bedeclin R&D. And the Pure R&D is basically shown here, where you have some people from London School and some people from J&J. With that, I will stop here and we can take any questions.
Thank you, sir, for such an informative and insightful talk. Now I invite participants to post their question in the chat box. So there is one question. What specific structural features of the microbacterial PUF-F did your team exploit with JNJ 6640 to avoid toxicity in human cells?
What was the question?
What specific structural features of the microbacterial PUF did your team exploit with JNJ 6640 to avoid toxicity in human cells?
Yeah, it's you synthesize hundreds of the compounds and you try to see which compound is the best, and then you have the counter assays, which I showed you, the human puref counter assay, where you screen the compounds you are synthesizing on the mammalian enzyme and see that those you pick up those molecules which have the best selectivity. So that's how you design those molecules.
Sir, I have one question. Can you throw light on this? You talked about the concept of generative chemistry in AI, where you build new molecules against specific targets. So, what are the steps? Like, how do we go about it? You need to have some database or some molecules reported based on that AI does, or how what is the mechanism of generation of new molecules using AI?
So yeah, I mean, generative chemistry, they claim, it's still a claim. They claim that if you have a crystal structure of a protein, or
Mm-hmm.
even if you have alpha fold structure of a protein, and you have the binding domain of your target domain of that particular protein, like what exactly things will bind. So that will basically, you just need these two. You don't need an existing data set, but that's what the claim is. And based on that, if you know where the domain is, your binding domain is, you have the crystal structure. The AI models can create de novo new chemical matter. So that's what the claim is. But I heard it several times that it's very difficult. It's not that easy. The way we say it, if you have data sets which in that particular binding pocket, that always is better in terms of generating new chemistries. So existing data set always helps.
So, are there any softwares or tools which work exclusively on generative chemistry,
Oh
which do
yes,
not
yes,
require
yes.
data sets? So, say, can
Yeah,
you
yeah.
quote some example? Uh,
I
like,
don't
don't
I don't
need
have
data.
the names in my mind, but usually people have their own proprietary softwares.
Okay, all right. And I wanted to know the source of bedaquiline, like it was rational design or was it isolated from some microbe or
No,
like,
it was a matter. It was a program which was coming. We were looking for Helicobacter pylori in gastric cancers at that time.
Mhm.
So the molecules were, or the initial chemistry was defined in that particular program. So we just screened for TB, and there is one of the hits, what we call, came out of that particular campaign.
Okay. And so there are two more questions in the chat box. So there's a
You can
question that
ask any question apart from Bedeclin, anything in biopharmaceuticals or anything you want to ask on drug discovery and innovation, please feel free to ask.
okay. So one question is nowadays bedaquiline resistance in MDR TB strains has been found, which is due to the mutations in several chromosome genes, including ATPE, RV0677C, RV0678, and PEPQ. So what's
Mhm.
uh sorry, yeah. So so what's the most personalized therapy for such cases?
Yeah, that's still very difficult to treat those people who have emerging resistance to bedaquiline, because as I said, that bedaquiline is a central drug in these patients. And the resistance is through the efflux pump, which is called RVO678. There are very few people who have developed inhibitors for efflux pumps or trying to see how best we can mitigate it. So luckily, at this moment, the resistance due to efflux is less than 5%. In some cases, it's a little bit more than 5%, but globally, it's less than between 5 to 7%. So it's still 90% or 95% of the patients are still susceptible to bedaquiline. But yes, going forward, resistance is inevitable. So how do we challenge and how do we address is part of the R&D.
Can I have a question?
Yes, sir,
Yes,
please.
please.
Yeah. What do you say where the natural product drug discovery stands with respect to addressing the infectious disease problem?
Yeah, and they are very natural products is where all the medicines actually came from. So if we have the source of new natural products, and if we know how to deconvolute the activity of the natural products, I think it makes it's easy to do it. But the problem is basically how do we, in the mixture of the natural products, how do we identify the right compound? So that's what most of the people fail in the natural products. And also now the source of the natural products, because we have exploited most of our, not most, we have exploited a lot of our original natural product collections. So now we need new sources, like people are going into deep oceans, people are going into very exotic ecosystem or habitats. So it's just we are not sure exactly what to look and where to look, and how to basically identify the molecules out of hundreds of those mixtures, the right molecule.
Just further add to that, going places to extreme, that kind of appearance seem to suggest that those might provide novel sources. But we are encountering a very, very interesting phenomenon. The gold drug factory called streptomycin, is it's very common, everyone will be aware of that the genus streptomycin has contributed significantly. And people have been talking of repeat discovery of drug from streptomyces. Somehow we got a bug to streptomyces that seemed to produce metabolites, range of metabolites. Like if you go by activity guided purification, you see that if you isolate from TLC, after fractionation, if you are isolating 7, 8 molecules, all of them show good activity. And it is not, such compounds that come up. So kind of how come, the question is how come so many molecules are showing up as antimicrobial in that particular preparation?
Yeah, I can't tell you the reason because I don't know enough background around that microbial preparation. But yeah, the question is that new chemistries or original chemistries can still be coming from streptomyces, or new methodologies in terms of culturing streptomyces.
Right. And what do you think should be the consideration and approach where the new drug that you may discover would have less chance or lesser chance, for that matter, of inducing resistance in the pathogen?
I think resistance is the misuse of the drugs what we have. If you look at in India, most of the antibiotics what we have in India are so misused that resistance is emerging to every new antibiotic class. So that's why Bedeclin excess is so strictly controlled by government across the world. So it's you won't go and buy the medicine in the shop. You can only get it through national TB programs. So it's very, very controlled. So the moment you have control and proper monitoring, then you have better chance of preventing the emergence of the resistance. Resistance emerges because we misuse the drugs, we don't complete our treatment courses, we just pop in a pill, and just because we think that it's without really having the need for that particular antibiotic at that particular stage. So yeah, and that's why the access to Bedeclin as a drug is so controlled, not only in India but also across the world, because everybody knows that this is such a precious drug, so it has to be controlled.
Yeah, correct. This is not the drug in the movement of resistance. My question was, can we have some strategy where, while discovering the drug or designing the drug, whereby the chance, like mostly what happens, either the efflux pump the different mechanisms of AMR. So could there be a strategy that these mechanisms can be bypassed somehow with, still?
Yeah, yeah, people have been looking at like developing drugs to block efflux pumps. So there have been a lot of programs, lot of activities on how to block the drug efflux, but it's not very easy. There have been not many successes in identifying the right molecules because there are so many efflux pumps, whether in the eukaryotic cells or whether in the bacterial cells, and different classes, different pumps, different mechanisms. So yeah, it's not that easy till now.
Yeah, thank you.
So someone has asked any specific AI algorithms, strategies for TB diagnosis and treatment?
I don't know. I am not in diagnostics, so I can't tell you.
For this treatment, AI?
Yeah, means for diagnostics everywhere, because if you uh, look at chest X-rays, let's say, on TB patients, or sputum smear microscopy in TB patients, because these are image analysis. So a lot of AI tools are coming into that particular thing. So imaging, what we call, and imaging is the one of the easiest thing to do from an AI perspective if you have a large and database of those images. So yeah, this is happening in terms of diagnosing and in terms of identifying what kind of TB is, especially the chest X-rays. If you have them, it becomes easier using a AI-based imaging tool.
Any other question we have? The participants can either ask directly or write in chat box. But I don't think anybody has question now. So my only thing is that people are talking about AI in healthcare. So diagnostics is okay, but what about from treatment perspective, like discovering new drugs and all?
Now, I
So
told you
that's
the
why I
generative.
asked you about generative chemistry. But
Yeah,
when
generative chemistry is there. You have now AI based data science design of the clinical studies. That's also something which is also very much coming up. That it's not only the discovery part where the AI is will be very helpful, but also in across the whole process of discovering and developing drugs. Like how do you select the right patient population, the biomarkers, the image analysis, everything will be more and more automatized. And that's where the AI is coming away. So it's not only the generative chemistry. I just showed you an example of AI helping even the selection of the clinical sites. How do you select the best clinical site? How do you select the best patient population? How do you select? You have seen in the oncology what we call personalized medicine. The first slide I show you, the Erome's law. Now it's getting better and better because of the personalized nature of the clinical studies and the trials what we do. So there are a lot of those things which are happening at different levels, whether it is recruitment, whether it is patient stratification, whether it's identifying the right biomarkers, whether it is uh, using the AI in clinical readouts, whether it's the AI in even in the preclinical and defining the potential tox associated with particular molecules. A lot of AI tools are there also. So yeah, so there is so much in every domain of drug discovery and development that AI is coming. And it's not only the small molecules, the antibodies. If you look at biopharmaceuticals, people even say that the way you are using the generative chemistry, you can use the antibody design for particular biological targets. So what we call de novo biosynthesis of novel antibodies. So there is a and I don't know how many of you have seen the Nature paper recently by this Nobel laureate who discovered that you can design antibodies on a computer against a particular thing. So these are called nanobodies, small antibodies, and this is all done in silico. Never thought about before. So yeah, so there is a lot of momentum across the whole thing, and it's just a matter of getting the right expertise and training yourself in those particular tools.
So there is one question. So Jyoti is asking, could you explain the trend of AI in discovering the mode of action of drugs, especially for natural products?
Mm, yeah, it's hard to say a mode of action because that's more of a complex biology. And so because that involves different tools, like whether you raise the resistance to the drug, or whether you do the biocore analysis, or what we call structural analysis, whether you do the cryo-EMs, whether you do any biochemical or biophysical assays. So at this moment, maybe within the essay itself, there are some components of the data science which comes into the play or at the instrument level. But at this moment, I don't think it is. Yeah, what we do from a mode of action, what we did is that we based on the scaffold. If you have 5 different chemical scaffolds, you can basically cluster different chemistries on their mode of action. So there's a paper published by us a few years ago on another project which was for viral drug target, where we showed that you can do the clustering approach in order to understand what a potential chemical matter can be doing. So I think it's on RSV, so you can have a look at it, and you can see how we did what we call clustering for understanding the mechanism of action.
So I think, sir, that's all. I don't think there are any other questions.
Thank you very much, all. And yeah, wish you a nice evening.
Thank you so much, sir, for enlightening us on latest trends in biopharmaceuticals, especially the AI thing and the journey of bedaquiline drug. And it was really inspiring and motivate us to work in this area. So
Thank
thank you,
you.
sir. Thank you, sir. Thank you.
Bye-bye.
Bye, sir. So with this, we finish the session. Today's session is over. And tomorrow again, we'll meet at 6 p.m. I will post the attendance link in the chat box for both the sessions, so you can fill the form.